1.5, which is fairly acidic. Okay, now that hydrochloric acid is produced by a cell type that are known as parietal cells. Okay, and they also produce a hormone, and I believe it is, yes, these blah blah blah, called gastrin. All right, and we'll talk about that in a second. Now, the second primary secretion of the cells in the stomach, secreted by what they call chief cells, is pepsinogen. And this is an inactive form of an enzyme called pepsin. And so what happens is when foodstuffs enter the stomach, we get this reflexive action, and hydrochloric acid is produced. It provides the acid environment necessary to change this pepsinogen into pepsin. And pepsin is proteolytic. In other words, it begins to break down protein. Takes these polypeptides and starts to break them down into smaller units, dipeptides, okay? So, carbohydrate digestion begins in the mouth. Protein, the chemical part of it, digestion begins in the stomach. All right, now there's another enzyme in there. Gastric gland, okay. They also secrete, there are cells in the stomach that secrete mucus. Now you got to think about this, regardless of what we eat, okay, we're chewing it, we're grinding it, we're mixing it with saliva and mixing it with mucus, not only in the mouth but in the stomach, because we want this material to move through the alimentary canal fairly easily. We want it to slide through. All right, so you have these cells that produce mucus to help coat the food and help it move through the canal. All right, pepsin, the most important digestive enzyme, and again, in its inactive form, it's called pepsinogen, and it is the acid environment provided by the hydrochloric acid that will change or convert this inactive form into pepsin. This is actually where they got the name for Pepsi-Cola, from the enzyme pepsin, okay? And this pepsin, its action is proteolytic, begins to break down protein. All right, now, the parietal cells also secrete this thing called intrinsic factor. All right, and what this substance does is help the body to absorb vitamin B12. And I think I remember seeing something about that. Aids in B12 absorption. B12 has an interesting name, if you've not heard it. It's called cyanocobalamin. C-Y-A-N-A, cobalamin. Anyway, this is absorption. This is important in red blood cell formation, vitamin B12. So there is a condition known as pernicious anemia. All right, and what that anemia, as the name suggests, has something to do with the blood. And what is happening is that this intrinsic factor is not being produced at an acceptable level, and that affects red blood cell formation. And so we become a little anemic. The number of red blood cells drops. They call that pernicious anemia. And that has to do with this substance.
So they just give you B12 injections?
They give you B12 injections. In fact, I've had those twice. For some reason, I don't know why, when I've gone into my primary, for a routine physical, my B12 is down. So, actually, he gave me the syringes and I injected myself.
For how long?
For 2 months.
Okay.
And then we go back and you check, and then the levels were normal, and then I didn't have a problem for, I think I've done that twice in the last 20 years. Don't know what causes it, but after a couple of injections, and I don't recall making any adjustments to my diet, but I may have or probably did, and then the problem, I didn't have the problem. Okay. So, from the stomach, the juices, the gastric juice, so-called, consists hydrochloric acid, gastrin, pepsinogen, intrinsic factor. All right, now, and we look at this gastrin. Okay, now, gastrin stimulates cell growth in the mucosa of the stomach and intestines, except where the gastrin is produced. And what this does is help to repair in the stomach lining, because we eat food that's it's not all smooth and rounded, and it's rough, and as it passes through, it damages cells, and those cells need to be replaced. All right, blah blah blah. We have another one. Cholecystokinin. Another component of gastric juice, and I'll break this word down so that we understand it. Cholecystokinin, if you look at that, this hormone action decreases gastric motility as the small intestine fills up. In other words, when food moves out of the stomach through the pylorus into the small intestine, and the small intestine swells, we get this stimulation of these cells in the stomach to produce cholecystokinin. Cholecysto refers to the gallbladder. Okay, and so what this will do, it decreases motility or movement in the small intestine, but it also, or in the stomach, and it also stimulates the gallbladder to contract. And the gallbladder stores bile. And so once we get this material that enters into the small intestine, and what bile does, bile takes, I guess for a simple way to explain it, it works on fat, and it takes big fat globules and breaks them down into little fat globules. And that process is called emulsification. So it emulsifies fat. You can liken this to after you've eaten a meal and you're washing the dishes and there was a lot of animal fat in the dish, and you see it floating on the top of the water, and you can take Dawn or whatever the dish soap is that you're using and squirt it in there on the top, and you can see that the big fat globules start to break up into little fat globules. And so that's what bile does. So when this material enters the small intestine, cholecystokinin decreases motility and stimulates the contraction of the gallbladder to release bile into the small intestine to break down the fat.
And bile ducts can become occluded, can't they?
Oh yeah, and they do with, and in fact, I had that. I've had my gallbladder removed because of gallstones.
You just made them, in other words, you kept making them, and all they had to do.
You know, most of the gallstones, you know, when they do an analysis, are, you know, they're cholesterol based, and they can collect. In fact, my whole gallbladder was filled with them, just like these little rocks of primarily cholesterol. And what happens is, I will take a look at the gallbladder sits up on the posterior side of the liver, and the function of the gallbladder is to store bile. Now the liver it produces bile, but when you're not engaged in digestion, in other words, there's not any foodstuffs in the small intestine, then that bile goes through the cystic duct, I believe, yeah, and is stored in the gallbladder. And then when you eat and the material enters into the small intestine and this cholecystokinin gets released, the gallbladder contracts, it ejects the bile.
Like a storage tank.
Huh?
Kind of like a storage tank.
It's a storage tank, yeah. Not the cystic duct, the hepatic duct, from the liver to the gallbladder. And then the hepatic duct joins the cystic duct to form the common bile duct, and that's what empties into the small intestine and empties the bile in there to break down fat. Because then you'll see, and we'll see that in a few moments, that there are enzymes, brush border enzymes in the small intestine, lipases, that will break down the fat further into its constituents, you know, the glycerol, the fatty acids, and what have you. But the function of this bile, and write that word down because you will see it, it emulsifies fat. And in essence what that is it takes the big fat droplets or globules and breaks them down into little fat globules, increasing the surface area so that the duodenal or the lipases can then act on those and break down those smaller fat globules into their constituents. Okay. All right, blah, blah, blah. Anything else we want to see there? All right. We have these accessory organs of digestion. All right, so we've gone from the mouth into the pharynx, into the stomach, and just review, you know, the secretions of the stomach, the hydrochloric acid, gastrin, pepsin, intrinsic factor, cholecystokinin. Just go back over them another time. And now we have an accessory organ of digestion that it's the pancreas. When I first began my career in nursing, I worked in the operating room up in Maryland, and one of the general surgeons there was Argentinian, and he took a group of the guys out one night to an Argentinian restaurant in Georgetown, and I had, for the first time in my life, beef pancreas. Didn't know what it was. It was delicious until I found out what it was. All right, but apparently they don't waste any part of the cow. All right, the pancreas, important accessory organ of digestion. All right. Some important enzymes. Well, we've already looked at the endocrine function of the pancreas with a previous chapter. The endocrine function, just to review real quick, we have the islets of Langerhans. Okay, we have the alpha cells, and they secrete a hormone called glucagon. And then we have the beta cells that secrete insulin. And both of these are involved in serum glucose regulation. Insulin lowers serum glucose, and glucagon raises serum glucose. And again, that's the endocrine function. Now, in terms of digestion, we're looking at the exocrine function of the pancreas. All right, so that means that these products that are produced by these cells in the pancreas will secrete their products, these pancreatic enzymes, into ducts, which will ultimately end up into the small intestine. All right. I won't go too much into the, you can look at on page 424 and 425, the diagrams of the pancreas and the gallbladder and the liver, just to note some structures, but let's identify the important pancreatic juices, if you will, and the digestive enzymes. And the first one that they address is called pancreatic amylase. AMALY, amylase. All right, and again, that pancreatic amylase, the function is to complete carbohydrate digestion. All right, so those disaccharides that were formed, or were the starch that was broken down in the mouth, when all of that material enters into the small intestine, the pancreas will release the pancreatic juice, if you will, into the small intestine, and a primary component of that is pancreatic amylase, and it completes carbohydrate digestion. And it's important to distinguish between which ones do what. The second is pancreatic lipase, and as the name suggests, this will complete lipid digestion. And if you remember, we had this bile that helped to emulsify these fats once they entered into the small intestine, and then these pancreatic juice will help to complete lipid digestion. You have nucleases, and these will break down, will digest nucleic acids like DNA and RNA. And we don't think about that particularly when we eat, but when we eat, a lot of times what we're eating, well, if we're eating plants, we're eating plant cells. And inside the nucleus of every one of those plant cells is plant DNA. When we eat animals, we're eating primarily muscle. And inside the nucleus of the multiple nuclei of the skeletal muscle of most animals is what we eat. Inside the nucleus of every one of those cells are nucleic acids. So we break those down, we break those DNA and then RNA into their constituents, which are nucleotides. And we use that plant and animal DNA to build our own DNA. Okay? All right. Now we have proteases that are called trypsin. Again, this is a pancreatic secretion, and it digests proteins, which, if you will recall, had begun in the stomach, and then the protein digestion will be completed in the duodenum or the small intestine by these trypsin, and we have chymotrypsin, all of these, and then carboxypeptidase. These enzymes are all involved in the digestion of proteins. Okay? All right. Blah, blah, blah.
What's the last?
Carboxypeptidase. And you'll know that it's involved in protein digestion because of the peptidase here. Proteins are held together, the amino acids are held together by peptide bonds. And you'll see these, nope, page before that. We don't want to get too much into that. Look before that.
Okay.
Yeah. We can just see these, the pancreatic juice, gastric secretions, and the carboxypeptidase.
Good, so that'd be page 423.
423,
Where it says
yeah.
pancreatic juice.
They split the bonds between some amino acids. And so all three of these, because now one by themselves is unable to break all of these peptide bonds between the amino acids of all the proteins that we eat. So we have these three that all work to break them down. Oh, and I do want to bring this up. And I brought it up here. I said that the nucleases break down nucleic acids, and the end products of nucleic acid digestion are nucleotides. The end products of protein digestion are amino acids. That's important. The end products of carbohydrate digestion are monosaccharides. Mono means one, and that being glucose, fructose, and galactose. All right, so that's the end products of carbohydrate digestion. The end products of lipid digestion.
Just a second. The product of blank are monosaccharides. What the product?
The end product of carbohydrate
Carbohydrates.
digestion are monosaccharides.
Okay.
These are what are absorbed into the bloodstream.
Okay.
These are what are absorbed into the bloodstream, as are these.
Okay.
All right, and then the last one, the end product of fat or lipid digestion, is primarily glycerol, and fatty acids. And again, we have to break these things down because these are what actually enter into the bloodstream, glycerol and fatty acids for the lipids. All right, so just, if you remember, mono means, when we're talking about carbohydrate, mono means one, and saccharin or saccharide means sugar. So monosaccharides are simple sugars. And these are what enter into the bloodstream. And again, for fat digestion, glycerol and fatty acids, these are what enter the bloodstream. Nucleic acids, nucleotides, and then for proteins, we have the end products are amino acids. All right.
Is that important for testing or
Yes.
is that important for general knowledge?
Both, but for testing, absolutely. I make sure you know the end products of these, the digestion of these materials. Okay. All right. Now, also from the pancreas, there is another substance that is secreted, and it is called bicarbonate. HCO3. And what this is an alkaline substance, And because if you think, now in the stomach, we have this high acid environment, very low pH, 1.5. Now, fortunately for the stomach, it has these cells that produce a thick layer of mucus to protect the stomach from this acid environment. Those cells and the number of those cells are not present in the small intestine. So the material, when it leaves the stomach, the chyme goes through the pylorus and enters into the small intestine, is very low pH and potentially very damaging to the cells. So the pancreas secretes a substance called bicarbonate to neutralize the acid chyme that's entering in from the stomach. Okay, you know what an ulcer is. You've heard of people that have ulcers. Two basic types. One is a gastric ulcer that occurs in the stomach, and one is a duodenal ulcer that occurs in the duodenum. Okay, for a variety of reasons. The ones in the stomach are primarily caused by a bacteria. Helicobacter pylori, yeah. But anyway, this bicarbonate that's secreted by the pancreas is controlled, the release of this bicarbonate is controlled by a hormone called secretin. Okay, and that secretin, is as the acidic chyme enters into the duodenum, the pancreas releases this, it's actually an endocrine function, releases this secretin into the bloodstream, and then the secretin travels throughout the bloodstream and then stimulates the pancreas to release this bicarbonate, okay, to help to neutralize the acidic chyme that's coming in there. All right. We got about 20 minutes. All right. So much for the pancreas. Again, look at the structure of the pancreas and its proximity to the duodenum, and you can see the pancreatic duct where it enters into the duodenum.
That's on page 424.
That is on page 424, yes. And you can see also in close proximity there the gallbladder, which has the cystic duct, the hepatic ducts that join to form the common bile duct, and then again you can see where that enters into the duodenum as well. All right, moving on, and I'm going through it rapidly. We'll look at the liver, an accessory organ of digestion, among other things. All right. Liver has a variety of functions, can't live without it. We're concerned primarily right now with the digestive function. I'm not going to go too much into the structure, but just do note where the gallbladder is on the posterior surface, the posterior bed of the liver. And blah blah blah. One of the some of the digestive function involved in there. One of them is we store. It stores glycogen. Okay, which is in layman's terms is animal starch. And this, when our blood sugar gets too low, the pancreas, the alpha cells will release that hormone called glucagon, and glucagon will enter into the liver and break down this glycogen into its constituent glucose molecules and raise blood sugar. Okay, this glycogen is also stored in muscle, but right now we're just kind of talking about the liver. All right, so it stores glycogen, it produces bile, we're talking about the digestive function. It stores vitamins A, D, and B12. And A and D are fat-soluble vitamins. Anything else we want to say about, the liver does a lot of things. The most vital liver functions concern protein metabolism. They include deaminating amino acids, and I'll explain. All right. If you remember what an amino acid looks like, the basic structures, you have a central carbon, you have a central hydrogen, and an R group, which is unique among each of the 20 amino acids. Then at one end of the amino acid, you have a carboxyl group, and then at the other end, you have what's called an amino group. These are just functional groups in organic chemistry. And when they say deamination, what that means is removing this amino group from the amino acids during protein metabolism. Okay? And then you can see what we're left with is the remaining part of the amino acid. It can do this to, your liver can make 12 of the 20 amino acids that exist in nature. In other words, it can deaminate one and then build another one out of that remaining material. The other 8 amino acids, you may have heard the term, they're called essential. You ever heard that term?
Yeah, sure.
Yeah, well, what that means is these are essential amino acids. It means that the liver can't make them, and they have to be taken in by diet.
You see those on foodstuffs.
Foodstuffs, cereals, for as an example. I don't know how true that is, but nonetheless, it gives them a selling point. So, liver stores glycogen, produces bile, stores vitamins, deaminates amino acids. It transaminates amino acids. That's building of amino acids. All right, so it's very important in the metabolism of proteins and amino acids. Your liver enzymes, if you've ever gone and had a complete physical workup, one of the things that they will check are your enzymes, which will be elevated if you have liver disease, like hepatitis A, hepatitis B, hepatitis C, or D, E, or F for that matter. But these transaminating enzymes, these break them down and these build them up, okay? And again, we are able to make 8 we're not able to make, and those are the essential amino acids. All right, not so much about bile. You can look at on table 15.3, just the major functions of the liver. Carbohydrate metabolism, when we're talking about, I do want to bring that up. When I said it stores glycogen,